Hello and welcome to this video on antivirals. Antivirals are one of the more important medications that exist. While not used as regularly in comparison to antibiotics, antivirals are still important, and they treat diseases like HIV, COVID-19, viral meningitis, and more. Like antibiotics, antiviral medication is either specifically tailored for a certain virus or broad spectrum. As opposed to antibiotics, most antivirals are not likely to cause harm to the recipient in terms of off-target effects or severe complications, although to be clear, this can happen, it's just not as common. Viruses come in two main varieties, RNA and DNA based. On these again, you have single or double-stranded genomes, but this is less important than what kind of nucleotides we're talking about. Viruses such as the pox virus, for example, herpes, and so on, all contain double-stranded DNA. When the DNA enters, it enters as and this allows it to slip into the DNA and create what you can think of as a insert that makes the DNA think, oh, we need to fill in the blank, and now you have two copies of it. When the DNA enters the cell, it's able to use the cellular machinery to then create new viruses by copying that. RNA viruses include things like influenza, measles, and mumps. RNA viruses don't enter the cell center in the same way that others like DNA viruses do. Rather, they sit either outside the nucleus or close to, and they use the cellular machinery there to make copies of the RNA. And this is done through a reverse transcription process, which takes the RNA and turns it into DNA, and the DNA then creates more RNA off of that template. The fundamental process of viruses involves infecting a host, and this is done by integrating the DNA into the host genome or by copying RNA in the nucleus of the cell. From this, the DNA or RNA is copied repeatedly, and these same genetics are also used to create proteins and more. These products are then used to create new virions along with the DNA or RNA that was created. By targeting the genetics, process, integration, or viral products required for the virus to survive, antiviral treatment can reduce recovery time. In the worst case scenario, you may only be suppressing a virus, such as with HIV, but given the consequences of not suppressing it at all, that is a significant improvement. Generally speaking, anything is better than death. There are a range of viruses and consequentially antivirals that exist, and we'll go through these in several steps. So if there's a particular topic or kind of antiviral that is of interest, you'll find timestamps listed below. The kinds of treatments and antivirals include acyclovirs, valacyclovir, penciclovir, famiciclovir, foscarnet, ribavirin, lamivudine, amantadine, and rimetadine. There's also interferon alphas, remdesivir, and nitric oxides. And yes, these are all a pain to pronounce, but importantly, we can address what each of these does individually, which makes it a little bit easier to understand what's going on, as the names do tell us something about what's happening. Acyclovir is a substitution, effectively. By having a drug that affects the acyclovir triphosphates, you're able to put barriers in the way of the virus integrating into the genome, and this is done by interfering with the phosphotransferases. Most commonly you find these related to cytomegaloviruses. By taking the phosphates and making it harder for the DNA to enter into the genome of the host, you can't duplicate the DNA. In some cases you can completely stop it from integrating. Valacyclovir is, in a manner of speaking, similar, although not quite the same. The change is only a small adjustment to the nature of the action, and all it really does is modify a side chain. But importantly, modification of that side chain has the same effect. It slows down or prevents the integration of the viral genome. Penciclovir does the same sort of thing again, but again we're looking at minor modifications to the side chains, but the results are again very similar. It prevents the DNA from having the ability to accept or for the virus to integrate with the DNA, and this means that you don't get the necessary adaptation from the cell to create new viruses by accepting the integration of that viral genome. In the best case scenario, preventing the virus from being able to enter the genome is the best possible option. And this is because, well, if it can't get into the genome, it can't have any real ability to survive. The RNA or DNA will eventually just be destroyed by the enzymes within the cell itself. This is where the next possible grouping comes in. Foscarnet is the first example, and this is a inorganic pyrophosphate. What this does is that it prevents the DNA strand from being expanded, and this is done by limiting the availability of what is effectively an essential product for the DNA polymerase to work. If it's not available, you don't have the opening of the DNA strand, and without opening the DNA strand, you can't have the insertion of any random new genome from a virus. This would be the next option, but of course this also has other issues with this cell's ability to, well, actually do things it needs to do, in particular with the DNA polymerase. If the problem can't be addressed with either of these options, which have effectively prevented the virus from being able to create more viruses through integration or use of the DNA's machinery, you have something like ribavirin. This is a drug used to substitute part of the DNA strand. It's a guanosine that doesn't have the necessary purine circle or cycle, and because it doesn't have this, you can't get the proper translation. Because you're in a proper translation, the RNA that comes out of this is either going to be too long, too short, or otherwise non-functional. And because it doesn't function correctly, it doesn't matter if the virus gets integrated initially into the genome, or whatever viral material might be produced from the host genome isn't going to be functional, and therefore it's not going to be able to cause any more issues. Lamivudine is in a similar sort of grouping, in that it's a pyrimidine-based target. It was primarily made as an antiretroviral drug, and then once it gets into the cell, it was found that, well, it actually does something a little bit different to what was hoped for, because it gets converted. What's important with that conversion is that it interferes with the transcriptase, and it becomes an inhibitor. Because it inhibits transcription, therefore the production of RNA and other virus material, it prevents the increase in the virus population. It's particularly useful for things like HIV, which are long-term viral infections that like to hide in cells that are otherwise immune to the immune system. So far we've covered the ability to prevent integration, to slow down integration, to prevent or drastically handicap the production of viruses, and in the worst case scenario, slow down the production to a crawl and thereby prevent the activities that are needed by the virus to be able to do its job. If all of these have failed, and there are instances where that will happen, you may need to treat the individual, and this is where targeting the parts of the cell that are either vulnerable to the virus infecting the host or are essential to the virus being able to work within the host cell come in useful. Ammonitide hydrochloride is an example of this. Because of its weird structure and the way it seems to work in the human body, it blocks the channels that are needed for certain viruses. And not only does this prevent the virus from being released from the cell, but it also interferes with the cell's ability to regulate pH, which means the cell eventually self-destructs or undergoes apoptosis because it can no longer function correctly, or the virus content within it causes it to die off because it simply overwhelmed the cell. And next we have what are practically naturally occurring phenomena within the human body and part of the immune system, glycoproteins in the form of interferons, but that are made artificially to be able to, well, treat various viral infections. Things like interferons particularly help to, well, prevent integration or incorporation of the virus within the host. In other instances, it prevents the viral proteins from being able to incorporate, and this means the cell membrane of the host cell doesn't have the ability to become the new virion envelope or similar. Now we have drugs that target RNA particularly, and the most common is going to be something like remdesivir. This is one of the older antivirals that we have, although it is still relatively new. What it does is that it targets RNA polymerases, And because of that, it's able to reduce the replication of the viral genome. And because you don't have replication of the viral genome, the rate of infection within the host goes down. This gives more time for the immune system to clear it out of the host and thereby prevent them from becoming infected or remaining infected. Unlike the previously mentioned examples, this isn't going to be targeting something specific to viruses. Rather, it's targeting what the host cell does normally. And because the virus hijacks and takes advantage of these mechanisms, by targeting those, you're able to practically counteract or undermine the attempts to, in turn, undermine the cell's biology. Although we can target parts of the virus, for example, by using interferons, or by targeting cellular processes or integration of the virus, sometimes those just aren't going to cut it. And this is where drugs that target either the virus enzymes or the virion envelope itself can be useful. Things like DNA polymerases, RNA polymerases, and viral neuramidases. As a general rule, interfering with DNA polymerases are going to have off-target effects, and that's simply because they're kind of necessary for the cell to be able to function normally. This is one reason why DNA polymerases generally are looking at inhibiting something that's going to be associated with the virion rather than a more universal opening of the DNA strand. We've already mentioned things like the pyrophosphates, but there's also polyphosphates and similar. All of these are particularly useful. The reason they're useful is that what they do is not just make it more difficult for the virus to be able to integrate into the DNA itself, but also to be able to get what it needs out of that DNA when it wants to make the necessary virus material to be able to get out of the cell and do more to infect other cells or new hosts entirely. It is, to use a very simple analogy, initially you can lock a book away and prevent access, and therefore people can't put things in the book. But what they're wanting to do here is, rather than be able to let people put things into the book, they want to prevent people from being able to open the book and read what is in it, and the instructions to do what needs to be done. Either way, you prevent this cell from being able to perform its function for the virus, and therefore the virus has less production of necessary proteins, for instance. Of course, that's not going to matter in the end if, somehow or other, the virus is still able to get the DNA translated into RNA of some kind, but this is where the second line of defense, so to say, can be useful. If you have an RNA polymerase or RNA-mediated polymerase, and by blocking that, you can't turn the RNA into protein or similar, therefore slowing down not just the DNA translation, but then the translation of RNA into something else means you can hit both steps of the virus's attempt to create more products. Unlike the virus, targeting this sort of approach won't affect the host to the same extent in the terms of eukaryotes and to a certain extent prokaryotes. That's because there are some differences in the kinds of polymerases that come into play, and by targeting those that are particularly related to viruses, you're able to prevent the virus from doing what it wants to do. If you're then going to have to look at targets after this, your tertiary targets, rather than having your primary and secondary targets, the virion itself is possible, and neuramidases are a good option for this. What they can do is allow the virion, the envelope around the virus, to become a better target for the immune system. This can be done by targeting the necessary pathways to generate products that will go in the envelope and protect it from the immune system, or at least give it a better chance against the immune system, or you can look at actually interfering directly with the products that are required by depriving the cell of what it needs to make those. In other cases, you can make the surface of the envelope practically inhospitable to the necessary molecules, let's say. These molecules on the envelope are needed for other functions of the virus. By making them inhospitable, you can reduce the ability of the virus to be able to infect hosts. We've spoken about the integration side of things, and the genome itself and expression of that. We've spoken about the products on the outside of the nucleus, particularly around the shell and the use of RNA. But there are specific sort of functional or mechanical processes you can directly target. For example, inhibiting translational processes, purely the viral mRNA, or you can look at mRNA in general. You could look at the early polypeptide chains that are needed for viruses, things like the synthesis of viral DNA, the non-viral enzymatic processes, or even biosynthesis and assembly. Starting with messenger RNA or mRNA translation, mRNA is needed to create proteins. If you can't read the protein, for example, by interfering with those molecules that allow it to be read, you effectively prevent it from being able to produce any of the needed proteins for that virus. This can be done by targeting the necessary molecules to translate mRNA into products. By preventing these enzymes from working, you don't get any results, but remembering that this can also have some off-target effects, but ideally you wouldn't be on an antiviral long enough for that to be a concern. If, for whatever reason, you can't target the messenger RNA, you may look at trying to prevent the early starts to the polypeptide chains or the protein chains that a virus needs. And by preventing the beginning sequences of many of these proteins, they're not going to form correctly, and because they don't form correctly, they're no longer going to perform their role as they are meant to. It's like getting jigsaw pieces and cutting them into random extra shapes, and therefore whenever you go to assemble them in the puzzle, they simply don't fit in the puzzle the way they're supposed to. You can do this by either disturbing the production process, or alternatively by substituting different parts of the amino acid sequence. Substitution in this case is not using a different amino acid, or rather using a artificially created amino acid, which is almost functionally identical to the real product, but just slightly different, and enough so that although it will pass through the process of taking mRNA into protein, once it's in that protein, it will not act the way it's supposed to act, and therefore the protein will no longer perform the role it's meant to perform correctly. Going a step further into the cell and to the virus itself, you can also inhibit the synthesis of viral DNA. Remembering in the same way that you can alter or substitute for the amino acids in protein, to a certain extent you can impede DNA by doing something similar. Generally speaking, this is either by blocking polymerases, which is perhaps the easiest option, or by altering the necessary nucleotides within, which makes it unable to be read correctly, or more importantly, replicated correctly for the purposes of creating new viruses. If you simply can't make that work, then fused DNA is another option, and this is depending on which of the 4 nucleotides you're looking at, whether it's a thymine or uracil. It is possible to prevent the genome from being read at all, and this can be done to prevent expression of the virus itself from within the cell. If it's not possible to target the approach specifically to what the virus needs for this to happen, for example, interfering with the virus's DNA, you can instead target more broadly processes involved in DNA synthesis. Now, this is going to have more off-target effects, primarily down to the fact that you will be targeting things that aren't specific to the virus, and therefore the host organism will see some of these same results. This will have a much greater effect on the virus, but by no means will the effect on the individual be insignificant. If you prevent the DNA from being able to be opened for more broad purposes, you're able to prevent integration of the genome from the virus, or alternatively, reading the genome of the host with the virus integrated, and thereby preventing expression of parts of the virus. Then there is the ability to block the necessary cellular machinery and prevent the construction of parts of the virus that it needs within the insides of the cell. Both DNA and RNA will lead to the production of various cellular machinery, or more accurately, virus machinery, including things like glycopeptides. For the most part, most humans don't have glycopeptides in the same sort of roles or production that the virus does. and this means that when it's observed by the immune system, it's identified as a target to attack. This is one reason why the virus tries to keep them inside the cell until the last possible moment. If, however, the cell can't be coaxed by the virus into keeping them inside the cell, or more importantly, assembling them for the virus, it's not possible for it to be able to survive very long. And so if you inhibit the production of this, or the assembly of it, you're able to prevent the virus from surviving for very long. This means you can either target the creation of those parts as needed, again through either providing the material that is faulty, let's say, or by preventing it from being put together correctly. Essentially giving a guy a flat pack and telling them to do it themselves. You'll come back a week later and they will still be refusing to look at the instructions. Nonetheless, with this, rather than being a flat pack, it's a lot of individual Lego blocks that need to be built up into a much larger structure, generally speaking the membrane of the, virus. Generally speaking, this is slightly more effective as you're not going to be preventing the production of cellular machinery for the most part by the host cell, but you're still going to be interfering with some of the other production mechanisms. Overall, this is what and how an antiviral works. The idea is that you want to start by preventing integration and infection as much as possible. If that's not possible, then prevent the virus from being able to use that integrated genome or slow it down as much as you can. If you still can't prevent the virus from doing what it wants to do, then try and give it faulty or dodgy equipment and materials to build more virus with. If that still doesn't work, then try and prevent it from being able to use that material once it is built by preventing assembly. If all of that fails, then you have means of stimulating the immune system to recognize that a viral infection is happening and to attack it more aggressively with things like interferons. Thank you for watching this video. If you found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions you have below.